speaker, Dr. Billy Christensen. He's a professor at the University of British Columbia Fisheries Center and an expert in ecosystem modeling. Those of you who are in my course last term will remember his name as being he's one of the co-inventors of EcoPath with Ecosim. Also executive board chair of the EcoPath Research and Development Consortium, which USF is a member. Lead author for the UNEP Global Biodiversity Outlook 4 report, which is the main publication from the UN Convention on Biological Diversity, and well known for a couple of seminal papers in fisheries science, including Fishing Down Marine Food Webs, which he published with Daniel Pauly in 1998 in Science, and Towards Sustainability in World Fisheries in 2002. So I hope we're going to hear about the bright future of EcoPath 6 and some of the other tools he has for visualization of marine ecosystems. So Dr. Billy Christensen.
Thank you, Cam, and thank you for coming. I actually asked Cam, do you think anyone is going to show up Friday afternoon? And Cam said, trust me, they'll come. I must say you have I like the traditions here. It's pretty unique, and it's a good way of making sure that Cam was right for once in his predictions. I know. But apart from that, what I would like to do this afternoon is to give you a personal view of some of the developments with ecosystem models. So that's the first half of the talk. And then, because Cam insisted, I'm also going to talk about some of the recent developments with the EcoPath EcoSim packages system, or modeling approach, I would call it, that I'll be working with. No, modeling framework, I think, is an even better word for it, but that's what it has developed into. It's not a simple model anymore. It's a framework. I'll be talking about some few new modules that I find especially interesting and which I think we are going to see a lot of developments with in the coming years. And that's roughly what I will be talking about over the next hour here. And you probably noticed this, I have this nice screensaver behind me. I'm not going to be talking about screensavers because it isn't actually a screensaver. This is interface for a scientific model that you see here. We have, as part of modularizing this EcoPath approach, we have made it so that we can connect to a 3D gaming engine. And we can let the scientific software then dictate what shows up in this 3D gaming world. It's called Blender, the approach we're using. And we've actually had quite a bit of fun about playing with this. But that's not the topic of the talk here, so I'll instead turn to fishery science. Not that the other is not fishery science. It is. It's actually pretty unique. How do you get an idea about how ecosystems looked when we were not there? Like, how did the Grand Banks look a few hundred years ago? You can't get National Geographic to show us that. That's one of the reasons why we're interested in these kind of visualizations. But let me turn instead to fishery science. When I started out there many years ago, by now, it was with single species. Studying single species, I was a herring biologist actually the first years. And gradually we started realizing, and this is not the royal we, I can assure you, right, Steve? You were there as well. That no, actually you can get far with just studying individual species, but there are limits because fish eat fish, and it has to be incorporated. This is a part of the move that we'll be talking a lot about later on this talk here about ecosystem-based management. But fish eats fish, and we have to consider that. That became clear, certainly in the 80s, that was necessary. As soon as you've taken that step, you also need to take the next step about what does fish actually eat. So you need to start looking at plankton, benthos, and it becomes a whole food web. That's the next necessary step forward. And once you've taken that step, you also realize, no, there's more to it. The fish and the ecosystems are impacted by the environment. And one lesson we've drawn, which I will be talking quite a lot about in this talk here, is that in order to understand ecosystems, we have to understand all of these components. We can't just go out and say what has happened in ecosystem based on the fisheries, or based on measuring the temperature and in the water, or just understanding the food web. We need to incorporate all aspects of it. That's where we moved, certainly over my career, has been to get to that realization. That's where we are now. I will come back to that later on. But before we come back to the present, let me just go. Now I'm making things complicated for myself here. Before we come back to the present, let us just go to the past, how this all started. A real pioneer here was Raymond Linderman, back in the late 30s. He did his seminal work on a place called, was it Box? Yeah, Cedar Creek Bog in Minnesota. He studied this as an ecosystem. He studied the energy flows. He was the first one to really do that. Up to that thing, these studies have been largely focused on individuals and how there would be interactions between them. But what Linderman did was to add a thermodynamic perspective to it, and he started thinking about trophic pyramids, transfer efficiencies, how energy moved through the systems. And much of what he did back then really serves as a foundation for the research we've been doing over the last 70 years when we're talking about ecosystems and how we model energy flows in ecosystem. That goes back to Raymond Linderman. And unfortunately, he died 27 years old. He managed to publish his PhD, and he died very soon after that. So for the PhD students here, he's kind of sets the bar, make that kind of study, and you don't have to do anything. No, he is certainly remembered because he was a scientist of a certain caliber in what he did and the way he organized it, and the study that he did was really, really unique. And it serves as an incredible model for how we study ecosystems to this day. Now, it inspired a lot of things, including 20 years after his death, the first international biological program. We have had many since then, but the IBP from 1964 to 74 was the first real big international program. And you had modelers inspired by Linderman's work, and Linderman and his associates, I should say, his mentor not least. Their work inspired an enormous international program during this 10-year period. Much of which was lake and tundra systems and a few marine systems as well. But it was a major international effort that directly came from and was inspired by this work. It also led to follow-up studies after 74, big consorted studies of ecosystems, on energy flows in ecosystems. And here I will especially point out a study which was made in French Frigate Shoals in Hawaii, where over a 5-year period, they studied everything that moved and didn't move in this system. So they quantified the energy flows in the ecosystem. And after a few years, they realized we have all these fantastic numbers. We've been studying this. What do we do with it? I don't know if you've I guess you've never experienced that kind of studies before, where people go out and sample and then they start thinking about how do we make sense of those numbers? And well, as a modeler, of course, I know how you make sense of modelers. You hire a modeler to do it. That's what we do, of course. And what they did there was a young oceanographer called Jeff Polovina, just out of grad school. They hired Jeff to make sense of the numbers that they've been gathering over this 5-year period. And Jeff, he's a smart guy. He's still around. Probably many of you will know him. Jeff looked at the great model at the time, because this was when, again following up from IBP, studies were emerging that got the food web into fisheries. The food web, how do you consider that in fisheries? And there were especially two initiatives that we should consider here. One was another NIMS activity. Taivo Lavasto and his group up in Seattle were studying, what was it? Was it Bering Sea? I think it was a Bering Sea model, really complicated simulation model. So Jeff went up there to have a look, and he also went to see K.P. Anderson and Eric Usin at the Charlottenlund Castle and Danish Institute of Fisheries. And actually, it must have been the time when I was a grad student there, actually working with Anderson and Usin's and some of the model and some of the assumptions in that. That's how I got into modeling. And he looked at these two complex models, Usin's North Sea model and Lavasto's whatever it was, some strange name. He looked at these two and he realized they were way too complicated for him to use in the French frigate Shoals. So he he looked closer into the Lavasto model, some of the key aspects of that, and he said, well, if I can just make, say, a snapshot of the model. So he took out some core aspects of the Lavasto model, and he turned that into what became the EcoPart model, and published that in 1984. I know many of you know everything there is to know about that, thanks to a recent course here. But I'll give you just a few highlights, nevertheless. The key aspect that he picked out from looking at the Lavasto model was mass balance. If you think about the energy balance of a group, there has to be a balance between, on an annual basis, between how much energy goes into this group and how much energy is coming out from this group, be it a species or a functional group or whatever it is. There has to be this balance. So he put that into his little linear equation system. And if you can do that for one species, you can of course do it for all species in an ecosystem. And that's a smart thing that Jeff introduced to this. This also means that where we have incomplete information, I'm sure we've all been in that situation. Where we have incomplete information, you may be able to use information about a predator to tell you something about the prey. As in, there has to be enough production of small pelagics to meet the demand of the predators of small pelagics. So that's a constraint that we can put into this set of linear equations that you see behind me. That's what Jeff Borovina did. And he used that to then see how it would balance. It's a pretty neat approach, and what it gives you is basically a snapshot of the energy flows in an ecosystem. This is how it's constructed. This is a plausible picture of the energy flows in a certain system. It actually adds constraints. And I've reached the point where I would say if you work with an ecosystem model, big complex model, whatever, one of the steps that should be involved is to check that mass balance, because it helps constrain the parameters in the model. And that by itself is really worthwhile. So that was getting EcoPart into the IBP, and the history, how it relates to that was my purpose with this part here. But up to now, we've only been talking about snapshots. We also need to make the movie version of this, the dynamic version of it. How do we get there? Well, at the IBP, they started this. Actually, a new field in ecology was established there called systems analysis. And the people in the IBP, the senior people there, recruited a number of smart young people with quantitative background to analyze the ecosystems. This was a time when computers became available, and there were some really smart people hired for this that moved the field of ecology quite considerably through these analyses, including looking at the dynamics of marine fish populations. And unfortunately, the models that they were working with had some major problems. Now, these models are basically all Lotka-Volterra models. So are all the models we work with nowadays on predator-prey interactions. They all come back to Lotka-Volterra. Does everyone here know what Lotka-Volterra models are? They should? Or should I explain them?
Explain it to me. I'm involved.
Okay, good. Thank you. I was hoping he would say something of that kind.
Take his class.
In a Locomotor model, you estimate how much does a predator eat based on how many predators there are in the system, how much prey is in the system, and then you add a search rate. So there's a very simple equation that's behind that and all the other models we work with. The problem with the Locomotor models are, well, I can tell you where to work actually. You have a lot of prey and you have a few predators. Lots of food for the predators, so they eat of the prey, and then there's less prey and more predators, and gradually you can come into wild oscillations. That's one of the things that Locomotor models actually can do. But they tend to be really unstable. Two aspects especially. One is a kind of the dynamic instability, where when you have these Locomotor equations, especially on the really fast turnover things like zooplankton, you very easily get into things going absolutely crazy. There's a dynamic instability which is kind of an artifact. But another, and from an ecological point of view, serious point is that they tend to self-simplify these models, which boils down to you have 5 kinds of grouper snappers or whatever you have, or 10 or 20 kinds of them, you're trying to model them, and after a few years in the model run, you have one left. The smart one takes over and the other ones are kicked out. So self-simplification, that things die out, has marred these kind of models that started with a system analysis in the IBP. It goes back to there. This meant basically that many of these smart quantitative system analysts left the field and started doing other things because they could not get these models to work. One of them was my colleague Carl Walters. Carl Walters had, at this time here, we're in the early 90s now, he had just published the Bible, Hilborn and Walters' Bible, in which he says using food web models for management is hopeless. He built that on his background in this. He had really done a lot of models, dynamic models of ecosystems, and had, like everyone else, the problems that they were so unstable. As a modeler, what we did with those models was put in if-then statements, so we didn't have things go extinct. You have to keep them in there somehow. Find ways of not having the model self-simplify. It was patchwork. Then one day, summer, early 1990s, Carl was out on the lake in the interior of BC on a beautiful summer day, fishing with his son Will. He was 9 years old at the time. Many of you know Carl, but I don't know if you've been fishing with him. I've spent a lot of time fishing with Carl. And what you should know about that, there are certain conservation measures at play here. When you go fishing with Carl, you don't go catching. I don't know why it is, but catch rates are usually much higher if he's not on board. I shouldn't. Are you actually taping this?
It's already on Facebook.
Hi, Carl. But actually, Carl admits it. There's something to it. So can you imagine sitting there in this little dinghy out on a beautiful lake in BC and not catching anything? They're out there fishing for rainbow trout. Nice, big, fat rainbow trout. And they don't catch anything. So after a while, of course, Will gets a little bit impatient. He starts looking over the side, and he sees these nice, big, fat Daphnia, Cladosians, Daphnia pulix, beautiful fish food. Will is finishing his PhD in nuclear physics right now, like in this spring here. He's smart. So he asks his dad, who's also kind of smart, why don't the fish eat them all? That's a fair question, isn't it? He knows that the trout would eat Daphnia, beautiful fat Daphnia. You don't find them as fish. Carl, of course, gives the explanation that any biologist could give, including you here if you think a little bit. He says, it's because we are fishing for the big trout out here in the open part of the lake. The small trout eats the Daphnia, but the small trouts can't survive out here in the middle of the lake, so they are up on the beach where the water is too warm for the big trout. They only come out here at dusk and dawn to feed, and then they rush back again. Otherwise, they would be eaten in no time. That was his explanation, and it was so obvious. It fits with what we know, behavior. Anyway, a little bit later, Carl realized that what he had told his son there was actually something that's very profound, and something that was totally missing in our ecosystem models up to that point. That, yes, it's not enough just to look at the average concentration of Daphne and look at the small trout and say, oh, they have enough food. You have to think of the behavioral state as well. Just think of a coral reef for a few seconds. I'm sure you all, especially down here, seen these beautiful coral reefs with all the small pelagic fish, red antias or whatever, swarms of them standing there around the reef. They're all waiting for plankton to be attracted to them. The smaller ones are very close to the reef. The bigger ones, plankivores, are further away, but they're all at such a place where when that single barracuda comes by, they dive into the reef. It's better to be half hungry than to be the food of the predator. So they stay in protection. They are all of these plankivores, all of them are dependent on this plankton that's attracted to the reef. They don't just go out and get it. Better to live another day. Now what Carl did then was to incorporate this into the Locomotor equations in a very simple way, simply by saying prey can be in two states. Vulnerable to predation, that's when they're out feeding, or invulnerable, that's when they hide. The question is, how do they go back and forth between those two states? That's what he kicked in to the Locomotor equations. And that became what's known now as the foraging arena. It's a fundamental aspect of, well, actually not just of EcoPart EcoSim, but it's an advantage too, because Beth Fulton, while she was developing this, spent a lot of time, months with Carl in Vancouver. Am I right? Certainly I'm right about she was there. The implication of that is suddenly, once we put that in, the models start to behave. We could replicate things, we could get things, we could move things, we could keep 10 kinds of